Let's say you have a new piece of furniture, or if you possess the patience, a model set. You could attempt to assemble it yourself, but there's a chance that you overlook a piece or put it together in a less than ideal way, especially the more intricate the item is. Ultimately, it's probably just best to check the instructions. If that feels like admitting defeat, think of it instead as taking the lead of what is possibly the most efficient production unit for the last 3.8 billion years, the cell. Welcome back to the science scene. My name is Shelby Bradford and I'm the Geeky Goth, your internet immuno-emo kid and all-around science-loving weirdo. Today, we're talking about instructions and regulations, which may seem like the antithesis of alternative culture, but are incredibly important for cellular function. Enough linguistic philosophy. We're going to take a deep dive into how our cells coordinate their activities through a molecular instruction manual called DNA. Previously, we discussed what nucleic acids looked like and introduced the 4 bases comprising DNA and an extra one used by its cousin, RNA. We said that in certain places within our genome, the arrangement of these bases creates a message which can be used to produce a functional unit called a protein. We're going to go deeper into that process today with an overview about how it works and how it's regulated. Let's just say that by the end of this, doing that home organization you've been putting off is going to look like child's play. First, let's orient ourselves a bit to understand what we're dealing with. The human genome is really, really big, about 3 billion base pairs. These are spread across 23 individual strands of DNA called chromosomes that, in most cells in your body, come in a pair. If you took all of the DNA from one of these cells and laid out the chromosomes end to end, it'd stretch about 2 meters long, or a little over 6 feet. So how does all that fit in a space that you can't see with your eye? It's because cells are also world-class packing champs. These DNA strands get wound around proteins called histones. It's kind of like winding a spool of thread. Only if we wound all of the DNA up like that, it'd be a pain to get to the spot we wanted when we needed to use it. Instead, only short stretches, about 146 base pairs to be exact, get wrapped up at a time with the help of 8 histone proteins, 2 each of 4 unique histones. This DNA histone combo is called a nucleosome, and they stretch all the way down the DNA strand. Together, the combination of DNA with all of these proteins is called chromatin. But the nucleic acid compaction doesn't stop there. To make more space, our cells fold over these DNA spools and stack these nucleosomes together. It continues stacking and folding, sometimes so much that some of the DNA becomes so tightly bound it's effectively inaccessible, which we call being silenced or closed chromatin. That's right. In almost every one of your cells, you'll have a good chunk of it that is so compacted it's never even used. It looks like that pack rat behavior that compels you to hold on to that box of stuff you never use truly is in your genes. In contrast to this closed chromatin, DNA, which contains frequently used segments in a particular cell, will be much more loosely packed, and so is dubbed open chromatin. This type of DNA is able to be manipulated when it's time to be used. Which brings up a good question. How is DNA used? To answer this, let's zoom all the way in on a section of one of these chromosomes. Not all of our DNA gets used for instructions to make proteins. In fact, most doesn't, and it is called non-coding DNA. However, don't think that because it doesn't serve the anticipated purpose, it's useless. Non-coding DNA is important for so much regulation and coordination in our cells. We just don't have time to discuss it right now. Instead, we're focusing on the parts of DNA that do provide protein instructions. As you could probably guess, it's called coding DNA. The coding segments are called genes and have the sequence that leads to a protein. How this works probably seems simple. Read gene in DNA, make protein with amino acids. Even considering our 3-base to 1 amino acid code trick we mentioned previously, this doesn't seem like it should be a concept that stumps first-year bio students. Except there are a few catches. For one, we need to use special machinery to make proteins, but all of those are in the cytoplasm of the cell. Oh, yeah, by the way, our cell isn't just a bunch of stuff in a disorganized sac. It has compartments. One is the nucleus, where the DNA stays, and everything outside of the nucleus is called the cytoplasm. Those are the only compartments we care about today, but suffice it to say, the cell likes organization. Okay, so all of our protein-making stuff is in the cytoplasm, but the DNA is in the nucleus, and it's not allowed to leave. So we need some way to move the instructions out of the nucleus to the cytoplasm. Enter RNA, specifically what we call messenger RNA, because it's going to carry a message. Look, appreciate the simple and direct science naming systems when they happen. Although we don't have any protein-making machinery in the nucleus, we do have a couple of proteins with specific functions. One of those is to print off a short, sweet copy of our particular gene into RNA. This is called transcription. But how does this protein know where the message is? Let's take a closer look at what happens. The enzyme responsible for actually writing our RNA message is called RNA polymerase II. Please don't ask why it's 2. It's directed to a particular DNA sequence called the promoter that lies just before the start of the gene. Now, a lot more proteins are actually involved in getting this RNA polymerase to this spot. The DNA will even fold and contort to bring everything together. But we're just going to get to the main point, which is this enzyme makes us an RNA copy of our gene of interest. Although it works a bit more like how you used to create negatives from a roll of film when you were printing pictures. If you ever in fact have done that. My God, I am old. Midlife crisis aside, remember that DNA is built with 2 strands of complementary bases. So how transcription works is the RNA polymerase builds its message out of nucleotides, and it knows which one to use by pairing the desired nucleotide to its matching pair. So to create the message we want, it actually reads from the opposite strand of DNA from where the gene is. Then, because this is RNA, it also uses uracil in place of thymine. After transcription is complete, there's a bit of cleanup that occurs with this newly produced RNA, but we'll save that for another day. Carrying on, to leave the nucleus, this RNA message, or mRNA, is given a chemical tag at one end like an export sticker, and then sent on its way, never to be seen again. Wait, what? That's right, RNA is not stable, and the cytoplasm is a hostile place. I mean, why do you think the DNA isn't kept there? So we can consider all these messages as time sensitive. Thankfully, these mRNAs often don't have to travel far to their next destination. The next enzyme in this process is typically nearby. Except it's not an enzyme in the typical protein sense. Instead, what will be our mRNA reading machinery is a ribosome, which is made up of another type of RNA. Nonetheless, it will be the housing to convert or translate this mRNA into a protein. This job, called translation, will be accomplished with the help of yet another set of RNAs called tRNAs. These are the tools that will recognize a particular set of 3 bases for the nucleic acid to amino acid conversion. Each tRNA carries a specific amino acid determined by the 3-base sequence that it recognizes. If you ever used a cheap decoder toy to solve a cereal box game, well, you get the concept. In translation, the tRNA that binds to the triplet sequence currently being read on the mRNA delivers the amino acid that it carries to the growing protein chain. So, our ribosome moves along the mRNA 3 bases at a time, and our tRNAs rapidly find their matches and deliver amino acids, which attach chemically in the confined ribosome and emerge to be folded into a protein. And voila! That's how DNA instructs protein assembly. But how does it know which one to make? Let's rewind back a whole topic and a few bad jokes. Remember how I said a lot more proteins besides RNA polymerase were involved in transcription? Well, some of those are what we call transcription factors. They will recognize specific promoter sequences, attach, and serve as a landing site for the RNA polymerase. Some of these factors get directed to that sequence when they're activated, like with a phosphate being added. This usually happens in the cytoplasm from a signal that came from inside or even outside of the cell. And that's cell signaling, which is a whole different topic. So, how do these transcription factors know where to go? Well, we don't know exactly. However, from what scientists can tell, these factors are able to search through the 3-dimensional mass of our accessible DNA. Recall that a lot of our DNA is bound up and inaccessible, so that makes it easier. These transcription factors also are looking for a sequence they can bind to, which we can picture as this combination of a 3D puzzle piece that also has a charge like a magnet. And lastly, there's not just one transcription factor searching, which means eventually one of these will hit the right place and get transcription started. However, it's also not just transcription factors regulating this gene expression. Even in our accessible DNA, it's still bound to histones. There's only about 20 to 100 base pairs between each nucleosome. I can assure you that genes are longer than 100 base pairs. So, to use this DNA, it needs to be released from these histones. If you haven't been impressed yet, this is where things get wild. Unwinding DNA from histones is called chromatin remodeling and is done with aptly named chromatin remodelers. Some of these are proteins and complexes which will physically remove or replace histones in the nucleosome. Alternatively, chemical tags may be added to these histones to make them more or less inclined to hold on to the DNA. Sometimes, these remodelers ride along with the RNA polymerase protein complex, since moving histones is kind of imperative for gene expression to happen, so it simplifies the process to have them with that machinery. To make this even more impressive, DNA itself can even be chemically modified to increase or decrease the likelihood of transcription at a particular site. This aspect of the genome, all of these regulatory elements and modifications, is called epigenetics because these act like a code on top of our DNA code. If you think it's incredible that all of these proteins and chemicals can coordinate DNA usage, here's another mind-blowing fact. Many of these settings are inheritable between cell divisions and even offspring. This, of course, makes it even more relevant when we're thinking about genetics and inheritance, but we'll have to cover that another day. All in all, you could say there is a lot going on inside our cells, even just with our DNA. And it's happening all the time. It really makes you appreciate not having to be responsible for keeping track of it. Thankfully, all of this cellular coordination and function takes care of itself. And it's still just a snippet. Maybe you are feeling inspired to take some cues from our DNA and find your own unique way to organize your tasks. Hopefully, though, now that you have an understanding of how DNA is used and a feel for what the lay of the land looks like, you want to dig more into details. If so, like and subscribe to check back in as we explore more cellular mechanisms and more awesome science. As for me, I have a date with a model set, and I will definitely be checking those instructions.